My name is Julia Martinez, and in this presentation, I will be analyzing Trompette et al. And the role of dietary fiber and short-chain fatty acids in the gut microbiota. Just to shed some light on the background of this article, the field of gut microbiota metabolism and its influence on allergic airway disease and hematopoiesis is very important because it sheds a light on the relationship between the diet, the gut microbiota, systemic inflammation, in particularly conditions like allergic asthma. This research article seeks to understand how dietary fermentable fiber affects allergic inflammation in surrounding tissues, specifically the lungs, and the role of short-chain fatty acids, or SCFAs, in mediating this process. The authors investigated the impact of dietary fiber content on gut and lung microbiota composition, and they observed changes in circulating SCFA levels and demonstrated that SCFAs also influence bone marrow hematopoiesis and DC cell function, dendritic cell function, ultimately shaping the severity of allergic inflammation in the lungs. Their findings contributed to a deeper understanding of how diet and gut microbiota metabolism impact the systemic immune responses and allergic diseases, offering potential future therapeutic interventions of targeting the gut-lung crosstalk. Before this article, research in the field of gut microbiota and its impact on allergic airway disease and hematopoiesis was pretty much already progressing, but still lacked comprehensive insights into specific mechanisms. Previous studies, such as those by Lepol et al. 2003 and Arpaia et al. 2013, have explored the role of gut microbiota and dietary factors in modulating immune responses and inflammation, particularly in the context of allergic diseases. Lepol et al. Investigated how functional characterization of human receptors for short-chain fatty acids and their role in polymorphonuclear cell activation. They also identified specific bacterial species that conferred protection against airway hyperreactivity. Arpaia et al. 2013 explored the immunomodulatory effects of dietary fiber and its metabolites, particularly SCFAs, on regulatory T-cell responses in inflammatory bowel disease. However, the understanding of how dietary fermentable fiber and SCFAs specifically influence allergic inflammation in surrounding tissues like the lung was relatively limited before the present article was presented. This study builds upon existing research by elucidating the mechanisms through which dietary fiber and SCFAs shape the immunological environment in the lungs, offering valuable insights into potential therapeutic strategies for allergic airway disease. The urgency of the question being asked truly lies in increasing incidence of allergic asthma and related allergic airway diseases in developed countries, coinciding with changes in dietary habits, including the reduced fiber consumption. Understanding how dietary fiber and its metabolites, or SCFAs, influence allergic inflammation in the lungs is quite crucial for addressing this rising public health concern. The broader implications of this research is simply that it highlights the interconnectedness between the diet, the gut microbiota metabolism, and our systemic immune responses. The mechanisms through which dietary fiber and SCFAs shape allergic inflammation, the findings offer potential avenues for novel therapeutic interventions targeting the gut-lung crosstalk to mitigate severities of allergic airway diseases. This research underscores the importance of dietary interventions and microbiota-targeted therapies in managing and preventing allergic conditions, with potential implications for public health policies and future clinical practices. To begin analyzing the methodology of the study, I'd like to begin with establishing the experimental design. Mice were divided into groups based on their diets: a standard 4% fiber content, or the control diet, a low fiber content, less than 3%, and diets supplemented with either cellulose or pectin. All mice were exposed to house dust mite extract, or HDM, through intranasal administration to induce an allergic reaction. Inflammatory cell infiltration, cytokine levels, serum IgE, and HDM-specific IgG1 antibodies, goblet cell hyperplasia, mucus production, and airway hyperreactivity were all measured to evaluate the severity of the allergic reaction. Fecal samples were collected to analyze gut microbiota composition using 16S ribosomal DNA amplicon sequencing, in which weighted unifrac distances and principal component analysis were all used to compare the microbial communities. Quantitative PCR was also performed to determine the relative proportions of firmicutes to bacteroidetes at the phylum level. High-performance liquid chromatography, or HPLC analysis, was also conducted on the fecal and serum samples to quantify specific SCFA levels. To assess SCFA effects on the allergic airway inflammation, mice were then treated with acetate or propionate in their drinking water to assess the protective effects against allergic airway inflammation. Lung inflammation, cytokine levels, goblet cell hyperplasia, mucus production, and serum IgE and HDM-specific IgG1 antibodies were evaluated once again. To evaluate the SCFA receptors and the immune cell activation, wild-type GPR41 deficient and GPR43 deficient mice were treated with propionate to investigate its effects on the allergic airway inflammation. Flow cytometry analysis was also performed to assess immune cell activation markers and cytokine production. Just to sort of extrapolate what we learned from the methodology, to go over the statistical approach of what the study utilized, statistical significance was determined using appropriate tests such as t-tests, ANOVA, or non-parametric tests, depending on specific data distribution and experimental design. The results were reported as a mean standard error of the mean, or the SEM, or standard deviation, with p-values indicating significant levels. Potential confounds may include environmental factors, genetic variations among mice, and variations in microbiota composition at the baseline. Sample sizes vary depending on the specific experimental group and analysis, but adequate statistical power was ensured where possible. Strengths of the methodology include comprehensive assessment of allergic airway inflammation, detailed analysis of gut and lung microbiota, and the investigation of SCFA effects on the immune responses. Some limitations may include the use of animal models, which may not fully capture human responses, and potential complexities in interpreting microbiota data due to various factors including microbial composition. To dive right into the real meat of the article, there were 4 major results that emerged most outwardly from the scientists: the effect of dietary fiber on pulmonary inflammatory responses, the impact of dietary fiber on gut and lung microbiota, the effect of SCFAs, particularly propionate, on allergic airway inflammation, and enhanced hematopoiesis of DC precursors by propionate, which we will review in future slides. To begin with the effect of dietary fiber on pulmonary inflammatory responses, the study demonstrated that dietary fiber content significantly influences pulmonary inflammatory responses. The mice fed low-fiber diets exhibited increased airway inflammation, characterized by enhanced inflammatory cell infiltration, eosinophilic and lymphocytic infiltrates, elevated cytokine levels, increased serum IgE and HDM-specific IgG1 antibodies, goblet cell hyperplasia, mucus production, and airway hyperreactivity. Conversely, the mice fed high-fiber diets supplemented with pectin showed a reduced inflammatory response and improved airway resistance. Impact of dietary fiber on gut and lung microbiota. The dietary fiber content alters gut microbial composition, with low-fiber diets decreasing richness and diversity and increasing firmicutes dominance. High-fiber diets, on the other hand, increase the proportion of bacteroidetes and Bifidobacteriaceae families. SCFA levels in fecal and serum samples that were collected are proportionally increased with higher amounts of soluble dietary fiber and decreased with lower amounts. These are 2 graphics that go in tandem with the 2 initial results that I explained in the previous slide. 1C is a representative H&E stained lung tissue from mice on either a control or low-fiber diet. Inflammatory cell infiltration was increased in the airways of mice on the low-fiber diet. This inflammation was dominated by enhanced eosinophilic and lymphocytic infiltrates as seen in the lung tissue between the control and low-fiber diet. 1F, on the other hand, is a representative periodic acid Schiff or PAS stained lung tissue from mice on either the control or low-fiber diet. Histological analysis of the lung tissue sections from HDM exposed mice revealed increased goblet cell hyperplasia and mucus production in mice fed a low-fiber diet as seen in the stain. To get into the last two set of results that were extrapolated from the study, the effect of SCFAs, particularly propionate, on allergic airway inflammation, or AAI. Propionate supplementation reduces cellular infiltration into the airways, particularly eosinophils, and decreases IL-4, IL-5, IL-13, and IL-17A levels. Propionate's protective effect against AAI depends on GPR41 receptor signaling. Propionate treatment influences dendritic cell activation and reduces effector Th2 cell proliferation, contributing to a dampened inflammatory response. Number 4 goes over enhanced hematopoiesis of dendritic cell precursors by propionate. Propionate treatment was found to increase the number of common dendritic cell precursors and macrophage and dendritic cell precursors in the bone marrow. Lung DCs from propionate-treated mice exhibited a reduced capacity to reactivate effector Th2 cells, contributing to reduced Th2 cell responses in the lungs. As we saw 2 slides ago, these are 2 stained samples of lung tissue collected from mice in both conditions in terms of propionate reducing the inflammatory response. In Figure 4D, a representative H&E stained lung tissue was collected from mice treated with or without sodium propionate on day 5 after the last HDM challenge. In Figure 4E, the analysis of IL-13 mRNA expression levels in the lungs and representative PAS-stained lung tissue from the mice treated with saline or sodium propionate were also 5 days after the last challenge. The dampened inflammatory response in the presence of propionate was reflected in the histological analysis of these samples, which showed blatant reduced cellular inflammation, such as Figure 4D. Goblet cell hyperplasia, mucus productions, and levels of IL-13 in propionate-treated animals in Figure 4E. Furthermore, serum levels of total IgE and HDM-specific IgG1 were significantly reduced in both samples. Overall, throughout all 4 results we've established, the results highlight the intricate interplay between dietary fiber, gut, and lung microbiota composition, SCFA production, and the immune responses in the context of allergic airway inflammation, or AAI. The findings suggest that dietary interventions targeting fiber intake and SCFA supplementation could hold therapeutic potential in mitigating allergic diseases by modulating immune cell activation and function. However, further research is needed to fully elucidate the underlying mechanisms and translate these findings into clinical applications. To begin this discussion, it's important to understand what exactly the article contributed to current knowledge. The article contributes significant insights to the field by elucidating the intricate relationship between dietary fiber, gut microbiota, SCFAs, and allergic airway inflammation. It provided novel findings indicating that dietary fiber influences AAI severity through modulation of gut microbiota and SCFA production, particularly propionate, which acts via the GPR41 receptor to help dampen inflammatory responses in the lungs. New knowledge includes understanding the role of dietary fiber in shaping gut and lung microbiota composition, identifying specific SCFAs, especially propionate, as key mediators of the gut-lung axis in regulating AAI, and highlighting the therapeutic potential of dietary intervention and SCFA supplementation in managing allergic diseases. Some strengths I found were rigorous experimental design and comprehensive analysis of the immune response and microbial communities, clear elucidation of mechanistic pathways linking dietary fiber to SCFAs and AAI, and further implications for potential dietary interventions to mitigate allergic diseases. Some limitations involved reliance on specific animal models to limit direct extrapolation to human physiology. Potential complexities in translating these findings to human dietary interventions, and subtle lack of exploration into the long-term effects and clinical relevance of these inflammatory responses in human populations. I think in the analysis of this study, it's important to consider some possible next steps for what this new information could lead to. Conducting clinical studies to validate findings in human population, investigating the role of dietary intervention and microbial modulation in managing allergic diseases in the future, such as clinical settings, exploring additional signaling pathways and immune cell interactions influenced by dietary fiber and SCFAs, and further examining the impact of dietary fiber on possibly other inflammatory diseases beyond AAI, expanding the scope of research in the field. These are simply some discussion questions that came to my mind when I was analyzing this article personally, having to do with future implications and also addressing some of the strengths and limitations that I discovered in the study. Personally, this study was very interesting to read as someone who struggles from chronic asthma, that there's a possibility that my dietary fiber intake could actually be heavily influencing how much of allergic reactions that I have to operating around my normal environment. The study and the research provided valuable insight into potential therapeutic avenues for managing allergic diseases, and moving forward, further investigation into clinical relevance and long-term implications of dietary interventions in human populations, it needs to happen to translate these findings into effective strategies for combating allergic disorders or possibly other disorders that could be alleviated by more research into the field.